Welcome to the lesson on DNA replication. Before we jump right into DNA replication, let's review the structure of DNA. This is DNA. DNA is a nucleic acid, and nucleic acids are made up of monomers or repeating units known as nucleotides. Here is a single nucleotide, and a sequence of nucleotides make up the DNA. Each nucleotide is composed of 3 pieces. This little circle here is a phosphate group. This blue portion here is a 5-carbon sugar called deoxyribose. And this last piece over here is a nitrogenous base. Nitrogenous bases come in 4 flavors: adenine, thymine, guanine, and cytosine, but we often call them A, T, G, and C for short. Cytosine and guanine base pair with each other, and adenine and thymine base pair with each other. You want to think of DNA as looking like a ladder, and the outside part of the ladder is made up of a sugar-phosphate backbone. So sugar, phosphate, that makes up the outside of the ladder. The rungs of the ladder, the inside of the ladder, are made up of these base pairs. The 2 DNA strands, because there's a DNA strand here and one here, are held together with weak hydrogen bonds. This whole ladder ends up being twisted into a structure known as the double helix. Now that we reviewed the structure of DNA, let's look at DNA replication and why it's even important. For most of its life, a cell has one copy, one set of its chromosomes. However, when the cell is getting ready to divide, it needs to make a copy of all of its DNA because each daughter cell needs its own DNA. When does the DNA get copied? You already know the answer to this. It happens during interphase, specifically during the S phase of interphase. If you remember, S phase stands for the synthesis phase because DNA is synthesized. DNA is replicated during this phase. Chromosomes replicate in their entirety, resulting in identical chromosomes. In eukaryotes, these molecules are called sister chromatids that remain attached at the centromere until cell division. So we have the sister chromatids, and we discussed how these are actually copies of each other. So the copies of DNA are held together until we're ready for cell division to happen. DNA replication happens in 3 simple steps. So the first thing that happens is the DNA double helix has to be unwound, and then it has to be unzipped. And the enzyme that helps do this is an enzyme called helicase. And you can see the helicase right here has separated the 2 DNA strands from each other. So if you remember, the 2 strands were held together by hydrogen bonds, which were weak enough that helicase was able to come in and separate the 2 strands from each other. Where the 2 DNA strands are separated, the area looks like a Y-shaped structure. So if you can see that here, it looks like a Y. And that part is called the replication fork. The second thing that happens is another enzyme is going to help out, and that enzyme is called DNA polymerase. If you want to look at this word right here, polymerase, to figure out what this enzyme does, it begins with polymer. And if you remember, polymer is actually a long chain. So polymerase, and if you remember ASE, ASE stands for enzyme. So this is an enzyme that's going to help us build a polymer. So DNA polymerase is going to help us build a strand of DNA. And how it does that is it's going to bring in new nucleotides. So here's the unzipped parent strand of DNA or original strand of DNA. We can also call it the template strand. And DNA polymerase is going to bring in new nucleotides and add it in to base pair with the template strand. So for example, right here is a little red nucleotide, and that stands for adenine. So DNA polymerase is going to bring in its partner, which is thymine, because A goes with T, and it's going to add that in, and it's going to keep moving up the DNA strand, adding in new nucleotides. So you're going to form another strand right here. Same thing is happening on the other side. DNA polymerase is adding in nucleotides right here and forming new base pairs. So a new strand is being built in this direction on the bottom. By the end of DNA replication, 2 identical DNA strands are formed. So this was our original DNA. We unzipped it using the DNA helicase. Here's the replication fork. And we built new strands over here. This is the direction of replication on this side, and here we're building the new strand on this side. By the end of DNA replication, we end up with 2 double-stranded molecules. Each double-stranded molecule is made up of 1 old strand of DNA, which is here in blue, and 1 new strand that we just built with the help of DNA polymerase, and that is green. Same thing on the other side, 1 green to represent the new strand, and 1 blue to represent the template or original strand of DNA. Because each DNA strand made at the end of replication has an old DNA strand and a new strand, we call DNA replication semi-conservative, because something is being conserved, that's the old strand, but something new is also being added, and that's the new strand of DNA. So DNA polymerase actually makes quite a few mistakes, about 1 in 2,000 base pairing errors, and you can see one right here. So this is good, A and T, G and C, A goes with T, and then right here is a mistake. T should not be base paired with C. This should be A. So that's a mismatch. That's a mistake. Thankfully, DNA polymerase has a proofreading function. So it's going to go back and check its work, and it's going to fix its mistake. And after DNA polymerase has done its proofreading, the new error rate goes way down to 1 in 1 billion. This is kind of like when you're handing in a really important essay for English and you do a rough draft. You don't hand in the rough draft. You proofread it, you fix all your mistakes, and what you hand in is much better than your rough draft. All right, DNA damage and repair. Unfortunately, there are things that damage DNA. There's certain chemicals that can damage DNA. There are things, types of radiation that damages DNA. UV rays, for example, X-rays can damage DNA, and they cause these mistakes to happen more frequently. Cells that are damaged have to be repaired. The DNA has to be fixed. And there are actually about over 50 repair enzymes that can remove damaged DNA and replace it with new undamaged DNA. And DNA polymerase, of course, helps along with that process. And an enzyme called DNA ligase helps glue back new nucleotides to help repair DNA. So we're always trying to repair damage. Sometimes, however, mistakes get through, and these are called mutations. Mutations involve any change to the DNA. Mutations get a really bad rap because they can be really bad. Bad mutations can lead to things like certain types of diseases, and cancers can happen as a result of mutations, but mutations aren't always bad. There are mutations that are neutral, right, that just cause differences in the population, right? For example, some people have blue eyes or they have red hair. Those originally started out as mutations that turned out to not be harmful. And then there are even mutations that are actually good. For example, humans were originally lactose intolerant. They were able to digest lactose only as babies. As humans got older, they became lactose intolerant. That was the normal thing. Around the time of Ötzi the Iceman that we talked about in class, a mutation arose where humans retained the ability to digest lactose after infancy. And that turned out to be a pretty good mutation because being able to digest lactose opens a whole new level of possibility to people, right? Because milk is filled with fat and calories, and that gave people a survival advantage. Other beneficial mutations involve things like resistance to malaria. There's another example where there's a subset of people who are immune to HIV, so they can be exposed to HIV, but they never get sick. And there's lots of mutations that are actually good. And we will discuss mutations even more later on, but this is just an introduction to them. So I hope that this was a good lesson that helped you understand DNA replication. Thank you so much for listening and watching.